Translational proofreading. Protein synthesis accuracy comes from charging tRNAs with correct amino acids. Proofreading is correcting translation by rejecting an incorrect amino acid tRNA before it can donate its amino acid. Protein synthesizing machinery achieves accuracy during elongation in two steps. First, proofreading. The accuracy of protein synthesis depends on two mechanisms: linking of each amino acid to its corresponding tRNA, base pairing of codons in mRNA to the anticodons of tRNA. So, any error occurs in the above two steps may cause damage in protein synthesis. Hence, to overcome this problem, it has two mechanisms to solve the errors. The mechanism is called proofreading. So, proofreading in translation: codon-anticodon base pairing. 16S rRNA forms H bonds with minor grooves of codon. Anticodon duplex only when correctly paired. Then mRNA 65' and 3' region, 60S mRNA correct pairing, anticodon loop of tRNA incorrect pairing, shown here. Proofreading in translation: incorrectly paired tRNA can't rotate into position or peptide bond formation. tRNA accommodation, correct base pairing, incorrect base pairing. Chemical proofreading: first mechanism used to improve accuracy of amino acid attachment to tRNA. Used to improve accuracy of amino acid attachment to tRNA. Used to improve accuracy of amino acid attachment to tRNA. It is done by the enzyme amino-acyl tRNA synthetase itself. Amino-acyl tRNA synthetase, which recognize an incorrect amino acid attached to tRNA and remove it by hydrolysis. When this enzyme binds the incorrect amino acid, it recognizes the different structure in its binding site and hydrolyzed and released that amino acid. Synthetase have two active sites: synthetic active site and editing site, hydrolytic site. Then kinetic proofreading: it is used to improve fidelity of codon-anticodon pairing. An incorrect tRNA molecule forms a smaller number of codon-anticodon hydrogen bonds than a correct one, therefore binds more weakly to the ribosome and is more likely to dissociate during the spread. Thus, the delay introduced by the elongation factor causes most incorrectly bound tRNA molecules to leave the ribosome without being used for protein synthesis. Post-translational modification. Next topic. The proteins synthesized in translation are as such not functional. Many changes take place in the polypeptides after the initiation of the synthesis, or most frequently after the protein synthesis is completed. These modifications include protein folding, trimming by proteolytic degradation, indene splicing, and covalent changes, which are collectively known as post-translational modifications. Polypeptide folding, proteolytic cleavage, indene splicing, chemical covalent modifications. Tertiary structure, pieces of polypeptide, position of the removed indene, new chemical groups are added. Post-translational modification in proteins. Translation, post-translational processing. Proteolysis, cleaving the polypeptide, allows the fragments to fold into different shapes. Then glycosylation, adding sugar, is important for targeting and recognition. Adding sugar is important for targeting and recognition. Phosphorylation. Added phosphate groups alter the shape of the protein. Then post-translational modification, proteins. Protein is there, ribosomes. Phosphorylation adds phosphate to serine, threonine, or tyrosine. Glycosylation attaches a sugar, usually an N or a O, in an amino acid chain. Ubiquitination adds ubiquitin to lysine residues of a target protein or for degradation. Sumanalization adds a small protein SUMO, small ubiquitin-like modifier to a target protein. Then disulfide bond covalently links the S atoms of two different types. Acetylation adds acetyl group to an N terminus of a protein or at a lysine residues. Lipidation attaches a lipid, which is a fatty acid, to a protein chain. Methylation adds a methyl group, usually a lysine or arginine residue. Hydroxylation attracts a hydroxyl group to a side chain of protein. Post-translational modification of protein. What? Post-translational modification refers to the covalent and generally enzymatic modification of proteins following protein biosynthesis. PTEM are important components in cell signaling and function. Types: phosphorylation, phosphate groups to specific amino acid residues on proteins. Glycosylation, carbohydrate is covalently attached to a target macromolecule, typically protein. So phosphorylation, phosphate groups to specific amino acid residues on proteins. Glycosylation, carbohydrate is covalently attached to target macromolecule, typically proteins. Methylation, addition of methyl groups to proteins. Acetylation, which is acetyl group from acetyl coenzyme A, is transferred to a specific site on a polypeptide chain. Ubiquitination, a post-translational modification, is typically achieved by E1, E2, and E3 enzymes that subsequently catalyze activation, conjugation, and ligation reactions respectively, leading to covalent attachment of ubiquitin, usually to lysine residues of substrate proteins. Then SUMOylation. SUMOylation proteins are proteins modified post-translationally and with small 12 kDa ubiquitin-like proteins called SUMOs. SUMOylated proteins are involved in various cellular processes such as nucleocytoplasmic transport, transcriptional regulation, apoptosis, and regulation of cell cycle proteins. Then meristolization. Meristolization is a lipidation modification where a meristonyl group derived from meristolic acid is covalently attached by an amide bond to the alpha amino group of an N-terminal glycine residue. S-nitrosylation. S-nitrosylation is the covalent attachment of a nitric oxide group to cysteine thionine within a protein to form a S-nitrosothiol. Proteolytic degradation. Many proteins are synthesized as the precursors, which are much bigger in size than the functional proteins. Some portions of precursor molecules are removed by proteolysis to liberate active proteins. Many proteins are synthesized as the precursor, which are much bigger in size than the functional proteins. Some portions of precursor molecules are removed by proteolysis to liberate active proteins. This process. Some portions of precursor molecules are removed by proteolysis to liberate active proteins. This process is commonly referred to as trimming. The formation of insulin from proinsulin conversion of zymogens, inactive digestive enzymes like trypsinogen to the active enzymes are some examples of trimming. Proinsulin, mature insulin, post-translational processing of insulin by proteolytic cleavage. Then intein splicing. Inteins are intervening sequences in certain proteins. Intein splicing. These are comparable to introns in mRNAs. Inteins have to be removed and extains ligated in the appropriate order for the protein to become active. Inteins have to be removed and extains ligated in the appropriate order for the protein to become active. Then covalent modifications. The proteins synthesized in translation are subject to many covalent changes. By these modifications in the amino acids, the proteins may be converted to active form or inactive form. Phosphorylation, the hydroxyl group containing amino acids of proteins, namely serine, threonine, and tyrosine, are subjected to phosphorylation. The phosphorylation may either increase or decrease the activity of the protein. A group of enzymes called protein kinase catalyze phosphorylation, while protein phosphatases are responsible for dephosphorylation, removal of phosphate group. Then hydroxylation, during the formation of collagen, the amino acids proline and lysine are respectively converted to hydroxyproline and hydroxylysine. This hydroxylation occurs in the endoplasmic reticulum and requires vitamin C. Glycosylation, the attachment of carbohydrate moiety, is essential for some proteins to perform their functions. The complex carbohydrate moiety is attached to the amino acids serine and threonine, O-linked, or to asparagine, N-linked, leading to the synthesis of glycoproteins. Vitamin K dependent carboxylation of glutamic acid residues and certain clothing factors is also a post-translational modification. Which one? Carbohydrate moiety is attached to the amino acid serine and threonine, asparagine, leading to the synthesis of glycoproteins.